Check, check, check. All right, we're a small group, but we're a mighty group. Thank you guys for coming in after lunch. Really appreciate you all. All right, it's time for our next speaker, a wonderful human being, so happy, so joyful. Please welcome to the stage the director of Immunomics. How do you say it? Immunome Center, the Buck Institute, Dr. David Furman.
Wonderful. Thank you so much. Yeah, I can see that. Well, thank you so much, organizers, for having me here. Thank you for listening today. I'm going to talk about something that has to do with everybody's lives, which is the progression of aging, how we progress in that aging trajectory, and how can we, you listen well, yes, it's good, how we can measure these as we go, as we, this is not the correct one, all right, as we progress in life. So one of the things I want to clarify as an overall arching goal is that we want to measure aging in every single step along the way. There is very clear hierarchical processes in aging. As you can see, we start from molecular and cellular changes, and that's what we call biological aging. That's where the hallmarks of aging fit into this idea of inflammation, cellular senescence, cell-cell communication issues, and so on and so forth. That starts at the age of 30, 35. That follows a functional decline, or rather phenotypic aging. And what is phenotypic is what we see. So if we do a scan, an MRI, we will see that as you age, there's a shrinkage of the white matter volumes, and that is phenotypic aging. You can also see that starting losing bone and muscle mass. That's all phenotypic aging. And that follows changes at the level of function, functional aging. So our hearing capacity, our cognition, things like frailty go later in life, and that's what we call intrinsic capacity or functional aging. And can we measure these things as we progress in the aging process? And so just to wrap up that idea that progression of aging starts with biological, follows phenotypic, and ends with functional before any disease kicks off. Okay, so this is biological aging. Let's start with biological aging clocks. Probably all of you in this room understands, or at least know about epigenetic clocks. These were the first ones that were invented in 2012 with Horvath. They're based on a DNA methylation and demethylation sites in the DNA that change with age so accurately that you can predict chronological age almost perfectly, right? Why do we do this? Because we want to predict disease risk. We also want to predict or evaluate interventions. If we're improving an intervention, you want to change that biological aging measurement. And so we've seen these type of figures in the past where if there's a deviation upwards, you develop diseases. If there's a deviation downwards, you're protected, you're healthy. This idea of the hallmarks of aging, I want to bring the concept of inflammation, right? It's in the middle, in the center, as a hub for all these hallmarks of aging. And can we measure that? Yes, we can. And we started the 1,000 Immunomes program exactly to answer that question. Can we find and identify biomarkers of systemic chronic inflammation that ultimately inform diseases and can be intervenable? And that's what we see here. We raised around 75 million from NIH. We wrote and published over 30 publications to identify biomarkers of systemic chronic inflammation. And we used AI and ML tools back in 2012 to derive this score for inflammation that we call inflammatory age. We published that in Nature Aging paper. And as you can see here, we can correlate that with multimorbidity, so the accumulation of diseases, frailty, immune function, ventricular remodeling of their heart, and this is exceptional longevity. And we can also predict mortality in external independent cohorts. Now, we run a clinical trial. It was a beautiful clinical trial. It cost us more than $5 million. It took us 2 years to complete. And I'm not going to go into the details of why and how the clinical trial was carried on, but all I want to mention today is that we took photos of the face of each individual in that study of the scalp as well, to see if that IH may be related to to wrinkles and so on and so forth. But then on my role as a chairman of the science board at Dior, we wrote this beautiful paper on skin aging and biological aging, and how all of the hallmarks of aging in the skin can be reflected in the body. And so I thought, well, maybe the skin is telling us something. Can we find a facial predictor of inflammation in the body? That's what we did. When we call that the healthy selfie program. And when my postdoc gave me this recognition, facial recognition, he gave me 37. I'm 49. He got a bonus. I was so happy. But then that's not just the prediction of chronological age. Now, can we predict biological age, inflammation in the body? And so that's Minia who did that. We can predict inflammation in the body. It's not perfect, and it doesn't have to be, because this can become a free tool for millions of people to use it around the globe, okay, and know pretty much where they are in their inflammation journey. You can see the accuracy is around 65%, good enough for a free tool, right, for diagnosing inflammation. I was going to China a couple of weeks ago, and I took that photo. I tested this healthy selfie, and my photo, inflammatory age, was not very beneficial. Maybe I was traveling, maybe I was stressed, and a lot of travel is not good for you. Bottom line. But that differs from your calendar age or the predicted calendar age. Anyway, let's move on to the functional pieces. Can we measure functional functional aging using these different clocks? If I had time, I would ask the audience who believes that aging should be considered a disease. I'm not going to do that, but only a few people. But in 2022, the WHO defined or recognized aging, not necessarily aging, but age-related decline in function as a disease in the ICD-11. So this is the International Classification of Diseases, ICD-11. Why is this important? Because now we can develop diagnostics and interventions and bring them to the FDA and get reimbursement codes, and it makes basically this free for everybody. For intrinsic capacity, I'm getting there. Yeah, 100% happy to talk after the talk. I have plenty of slides to show today. All right, so here we run another 1,000 immunomes, call it, in France. If you can just help me keep track of that, just thank you. That took 10 years to complete. And here we're looking at functional changes, cognitive status, physical performance, nutritional status, and so on. And we built yet another clock. But in this case, it's not a clock trained on calendar age. It's a clock trained on function. It's the first clock that defines how good you're aging in your functions in the body. And that has almost no overlap with previous clocks. So we're discovering new biology. The IH that I showed you before is the largest contributor to this clock. Anyway, it's a long story that was published again in Nature Aging. Let's see if that, oh, let's go back. People with high intrinsic capacity live 5 years longer. And if you have high content of omega-3 polyunsaturated fatty acids in your blood, chances are you have a better intrinsic capacity. Go eat your fish. Remember always to eat your salmon and herring. So anyway, so here you have the overall epigenetic age, right? I don't think it's a great tool when you look at those true age clocks and so on to understand the organ level aging. Because say you have age acceleration of your heart plus 5 years, but you have less, -1 for the brain and then -3 for the kidney. Overall, your biological age acceleration is 0, so you're doing great. Yet you have some weak organs. So can we find a way to predict organ aging at the level of those systems? Yes, we used 450,000 people to train this model. This is a mortality predictor due to different diseases. And that was validated in the health and retirement cohort. I'm going to go pretty quickly because I want to touch on the last piece. How do we build this clock? Nothing fancy, just using regular biochemistry labs. Okay, so if you have your labs, your favorite Quest or LabCorp, bring them to me. I'll just tell you where you're at. This is massive. This is a game changer in the area of precision longevity. I love this figure because it shows that it doesn't matter how accurate your clock is at predicting chronological age. It doesn't really correspond to a better prediction of morbidity, but that's a scientific detail. I don't want to be the scientist, the boring scientist in the room. Sorry, guys. All right, and you can also produce these clocks based on any omics, your proteomics, your metabolomics, your transcriptomics, your epigenetics. We can take those, plug them into an algorithm, and produce these disease age or organ age specific clocks. Just to add some more flavor, if you eat some white bread, you have age acceleration. For most clocks, hamburger not good, margarine, fish sticks, red wine. Don't ask me how and why. I'm not biased here. It is actually apparently driving deceleration across the board. Now, exercise, beautiful. You have a deceleration of most of the clocks, including metabolic, circulatory, not so much for the brain. And then if you're a smoker, yeah, that's you're not doing well. You have acceleration of all of the systems. First and foremost, respiratory. Last piece, I want to convince you not to go to the moon. Okay, so all right. So here you see some biological age clock. You see this person, you say, oh, that has this acceleration. Dang, we have to do something. You see this guy, well, it's all good. We don't have to care about. Now, if you look at the longitudinal data, 1,000 immunomes data, look at this guy. Started pretty high, but it didn't progress. It didn't actually, it was quite well over the years. This guy here developed cardiovascular disease. Okay, can we predict the future? Peter said no in the previous talk. I would argue you're wrong. We can with the test. So listen for just a moment here. We are looking at space at large. Astronaut health, we can take your cells, put them in microgravity on the ground, and have a window for your future 5 years of aging. So we can predict. We have one blood draw from you, put in microgravity, 2 time points, the current one and the predicted one. And that predicts 5 years out. So there's a whole lot of science behind. I don't want to, again, be this boring scientist in the room, trying to be a little entertaining here. But with the PBMCs, we developed this blood test. And that culminated in the formation of Cosmica Biosciences. I'm going to go quickly because I have only 1 minute. I want to just show you a snippet. Oh, we don't have audio here? Okay. No audio for the video. Oh, dang. Is there any audio there? No? No audio? Okay. Okay, this is discouraging. We only have 28 seconds, so I'm going to jump to the actual product we have, which is called Beyond Age. Can we move the next slide, please? There you go. Okay. This gives us the opportunity to have a pace of aging, a trajectory of aging. We created that. These are all the clocks that I alluded to, a summary of my presentation today, and just big thanks to my team. We do a lot of things with dinner and see the giants, and we have a lot of fun other than the hardcore science. Thank you so much for your attention. Right on time.
Perfect, perfect. I love this guy, man. He is so positive. Thank you, David.